So I can announce the last speaker of the session. It's Anna Maria Gambetta from the Department of Molecular Medicine, Medical School at the University of Padua in Italy. She is a PhD student, and she will give a presentation on a genome-wide screening in pluripotent cells, identifies MTF1 as a novel suppressor of mutant Huntington toxicity.
Good morning, everyone, and thank you for the opportunity of being here and share with you our study on Huntington disease. As we know, Huntington disease is caused by a mutation, and several biological processes have been involved in its pathogenesis. Unfortunately, we still don't know which one is the most promising to target for therapeutical approaches. For this reason, we decided to perform an unbiased genome-wide screening in order to find genes whose activity could protect cells from the detrimental effect of mutant Huntington protein. Our idea was to set up an in vitro HD model based on ES cells and then to perform a screening. The hit of our screening have then been validated in vivo and in vitro and in vivo, and such genes will represent the target for a gene therapy. This is our HD in vitro model, and as you can see in orange, the mutated cell line show a decreased cell proliferation rate, increased cell death, and increased ROS production. We decided to use this model for, as a platform for a screening in order to identify genes who can protect cells from these events. Our gain-of-function screening is based on insertional mutagenesis, meaning that if the cells bearing such mutation can survive against and protect against the effect of mutant Huntington, it will survive and form a colony. We performed a total of 17 mutagenesis experiments with more than 10,000 integration events. And after selection, we identified 107 genes, which are here represented in this network. As you can appreciate, they're highly interconnected, meaning that they're not just random surviving clones, but that they're part of a shared program. But who are these genes? Well, first of all, this network is highly enriched for HD-related genes and HD-interacting proteins. And moreover, we found no signature for cancer or apoptosis. Now I'm going to show you the validation of our favorite candidates, which is MTF1. MTF1 is a transcription factor that regulates metal homeostasis in the cell. It is known to induce metallothioneins, which are metal chelators. As you can see here in green, the overexpression of MTF1 can rescue cell proliferation, reduce cell death, and reduce ROS production. So after observing such a promising protective effect in vitro, we decided to test our candidate suppressor gene in vivo system. We started from zebrafish. We know that the expression of mutant Huntington leads to malformation and cell death in embryos. And we observed that with the co-injection of our candidate genes, MTF1, we could rescue the fraction of LT larvae and reduce the number of malformation. So prompted by these results, we decided to test our candidate also in a mouse model of HD, the R6-2 mice. So here is our gene therapy approach. It is based on adeno-associated viral vectors carrying MTF1. So these experiments have been carried out by Vittorio Maglione's group in Nørremølle, and we really thank them. And with this beautiful result, we can observe that our R6-2 mice treated with our vector containing MTF1 performed similar to the wild-type littermates in both of the behavioral tests. Moreover, we could observe a significant reduction in the number of aggregates and a reduction in ROS levels in the brain of those mice. So in conclusion, we developed an in vitro model for the identification of suppressor of mutant Huntington toxicity. So far, MTF1 is our best candidate, and we validated its protective activity in vitro and in 2 in vivo systems. And now we're ongoing with the validation of its activity also in a human in vitro system. I'd like to thank all the people who contributed to this project and all of you for your kind attention. If you want to know more, if you want to know more about our project, come and visit my colleague Sonia at the poster session number 10 on the ground floor. Thank you.
Thank you very much. Questions? Yes. Sarah?
Very nice talk, and I love that sort of screening. So as far as I understand it, MTF1's a transcription factor, isn't it? Involved in metalloproteases, I think. So I was interested in terms of that being a therapeutic target and what your thoughts are in terms of mechanisms, because it's very interesting data.
Thank you for the question. Yes, the mechanism is under investigation. Of course, we have some hypotheses. For example, we know that heavy metals promote mural antigen aggregation and ROS production. So our hypothesis is that maybe MTF1 has a mechanism which is ROS mediated, because we already showed that there's no direct interaction between MTF1 and mutant aggregates.
Very nice.
Thanks.
And you could look at it relating to somatic expansion as well, in terms of whether an oxidant damage, although it's debated, is promoting DNA damage in non-dividing neurons. And pertaining to what was discussed earlier, you could look at
But this is a very interesting suggestion, and for sure we will investigate also this field of stress. Thanks.
Very nice.
Thank you. Other questions? Last chance.
Thank you.
Okay, thank you, Anna.